Here are the best line summaries of FSC Biology Chapter 6 for the preparation of PPSC lecture and subject specialist biology and zoology. In this module, we shall discuss these major 8 topics from discovery of bacteria to cyanobacteria, and we shall also divide these 8 topics into smaller ones so that we may remember them with reference easily and we may link our concepts. Now let's start with the discovery. Pro mean before, karyon mean nucleus, and archae mean ancient. Please remember these meanings. Microbiologists divide bacteria into 2 categories, eubacteria and archaeobacteria. Who divide bacteria? Microbiologists into eubacteria and archaeobacteria. Microbes like bacteria and protozoa were first reported by Leeuwenhoek in 1673. Please remember this name, Leeuwenhoek. He used drawings in the microscope and then called them animalcules, small animals, not animals. Louis Pasteur developed vaccines for these 3 diseases, which were anthrax, fowl cholera, and rabies. He first of all proved that microorganisms cause disease, and that was a milestone in the history of disease science as well as parasitology. Germ theory of disease was formulated by Robert Koch, and this theory have 4 postulates. We will discuss it later. Robert Koch also isolated bacilli from the blood of anthrax-died sheep, the sheep which died of anthrax disease, and also TB and cholera bacteria. He also invented techniques of inoculation, isolation, media preparation, cultures, etc. Please remember all these techniques. These are very important, especially in microbiology. What is inoculation? Inoculation is the transfer of organism's colony from one petri dish to other, or from one petri dish to the body of organisms. Bacteria are found everywhere in hot springs and highly saline or acidic soils. Germ theory of disease have 4 points or 4 postulates. So if you want an organism to be declared as germ, he should fulfill all these 4 requirements. Organisms must be found associated with the disease, and if it is associated, it can be isolated and grown in pure culture. This pure culture produces diseases when inoculated. It should be recoverable from infected animal. Now, if any organism fulfills all these 4 requirements, that organism can be declared as a germ. Structure of bacteria. Bacteria range in size from 0.1 to 600 micrometer. Mycoplasma is the smallest genus of bacteria. Please remember that Mycoplasma is the smallest genus of bacteria, which is equal to largest virus, pox virus. E. coli is 1.1 to 1.5 micrometer wide and 2.0 to 6.0 micrometer long. Spirochete reach up to 500 micrometer, while Staphylo and Strepto are 9.75 to 1.25 micrometer. Epilopisium fischelsoni is the huge bacterium who live in intestine of surgeonfish. There are 3 types on the basis of shape. Please remember cocci, bacilli, and spiral. Other shapes may be trichome, forming sheathed, stalk, scare, star-shaped, spindle-shaped, lobed, and filamentous bacteria. Please remember all these shapes. Cocci. These are spherical or oval. Division in one plane produce Diplococcus or Streptococcus. These divisions are important to remember, and what they produce is important. Paired cocci are Diplococcus, while a long chain are Streptococcus. Division in 2 plane produce tetrad arrangement. Please remember, division in 2 plane produce tetrad arrangement. Division in 3 plane produce Sarcina. Sarcina is a cube of 8 cocci. This is very important MCQ. Sarcina is a cube of 8 cocci. Division in random planes produce Staphylococcus grape-like cluster. Diplococcus pneumoniae and Staphylococcus aureus are examples of cocci. Please remember the examples of cocci. Now bacilli and spiral. They are all rod shaped. all divide in one plane only to produce bacillus. There is only one plane, not the specificity of 2-3 planes. Only one plane. Streptobacillus is chain of bacilli. Streptobacillus is chain of bacilli, while paired bacilli are diplobacilli. Please remember that. E. coli, B. subtilis and Pseudomonas are example of bacilli. Now spirals. Spiral comes in 3 forms. Please remember this fact that spiral comes in 3 forms: Vibrio, spiralum or spirochete. Vibrio are comma shaped while spiralum is thick rigid spiral. Now very important and famous MCQs to remember: thin flexible spiral is spirochete. Please remember this. Vibrio and Hyphomicrobium are spiral bacteria. Flagella, which originate from basal body, which is present just below the cell membrane in the cytoplasm, are thin hair-like appendages. They are made up of flagellin proteins. Remember this, they are made up of flagellin protein. Which protein? Flagellin. Now these definitions are very important to remember. Atrichous bacteria are without any flagella. Monotrichous bacteria have single polar flagellum. Lophotrichous bacteria have tuft of flagella on one pole or side. Amphitrichous bacteria have tuft of flagella on both sides or both poles. Peritrichous bacteria have flagella around whole cell body. Cocci rarely have flagella. In addition to motility, flagella also respond to chemical, which is called as chemotaxis. Pili, smaller than flagella, not involved in motility or any kind of movement. Please remember that present in gram-negative bacteria only, made of pilin protein and involved in mating, which is conjugation. So they are used to transfer genetic material from one bacteria to the other, and they are means of attachment with surfaces. These are the references of pili with the flagella. Please remember all these facts because they are very important in the regard of exams. Now capsule, slime and cell wall. They are tightly bound to cell, made of either polysaccharide or protein or both. It is called capsule. So they are made of either polysaccharide or protein or both, and they are tightly bound. Capsule provides sticky characters to encapsulated bacteria and create pathogenicity. They provide sticky characters. Loose soluble sheet made of macromolecules are slime capsule. The only capsule are tightly bound, while the slime capsule are loose and these are soluble. Slime provide greater pathogenicity and protect them from being phagocytosed or being eaten. Cell wall protect bacteria from osmotic lysis or breakdown and is absent in mycoplasmas. Gram staining was developed by Christian Gram. Please remember, gram-positive bacteria are stained purple. They retain the primary dye due to CV1 complex. Gram-negative bacteria stain pink by retaining secondary dye. Now cell membrane, cytoplasm, nucleide. Bacterial cell membrane don't have sterols like cholesterol and have enzyme for respiratory metabolism. Please remember these two characteristics because these two characteristics are distinguishing characteristics which distinguish bacterial cell membrane from the other cell membrane. Bacterial cytoplasm lacks cytoskeleton or microtubules and organelles. Plasma membrane and cytoplasm of prokaryotes, these two collectively called protoplast. Please remember this definition which is very important. Bacteria has ribosomes and mesosomes. Bacteria has single, circular and double-stranded DNA in dense area which is nucleide. Nucleide is a stained region which is stain. Please remember this: single, circular and double-stranded DNA in dense area is called as nuclei. Bacteria are haploid due to single chromosome. They have single chromosome, so they are called as haploid. Nuclei become visible in light microscope by staining with Fulgen stain. Chromosome of Escherichia coli measures about 1400 micrometers. The structures in bacteria include plasmids. These are very important to remember, read, as well as understand. Plasmids are circular double-stranded DNA molecule. They are self-replicating, means they can replicate itself and make copies of themselves, and not required for bacterial growth and metabolism. Bacterial growth don't require plasmids. This is very important to remember. Plasmids have resistant genes against drugs, heavy metals, diseases, and insects. You can be asked what is the function of plasmid. So they have resistance genes against drugs, heavy metal, disease, and insects. On the other hand, they are very important in genetic engineering as vector. Prokaryotic ribosomes are smaller than eukaryotic, also attached with plasma membrane. So the important fact to remember that the ribosomes of eukaryotic cell are attached with the plasma membrane. Mesosomes may be vesicles, tubules, or lamellae. Now this is important to remember that mesosomes may be vesicles, tubules, or lamellae. They are involved in DNA replication. This is important function that mesosomes are involved in DNA replication and have respiratory enzyme. Common waste of bacteria are alcohol, lactic acid, and acetic acids. This is different from the waste of eukaryotic cell. Spores are dormant bodies and may be endo or exospores and resistance. What is the difference between spores versus cysts? Spores are produced during late cell growth, while cysts are formed during differentiation. Please remember these two. Spores produced during late cell growth and cysts produced during differentiation. Spores are heat resistant while cysts are not heat resistant. Cysts are only desiccation resistant while spore can resist light, temperature, pH, and chemicals. Cysts are only desiccated areas resisted. Nutrition. Most bacteria are saprophyte or dead eater or parasite. Saprophyte get their food from dead organic matter. These may be decomposer. They are called as decomposer. So bacteria are dead eater, so they are saprophyte. Humus. This is very important definition to understand and remember. Humus is partially decayed material of plants and animals in soil. Chlorophyll in photosynthetic bacteria is dispersed in cytoplasm. They utilized hydrogen sulfide instead of water as source of hydrogen and liberate sulfur instead of oxygen during photosynthesis. Nitrifying bacteria are chemosynthetic and oxidize inorganic compounds like ammonia, nitrate, sulfur, or ferrocene. Please remember that the nitrifying bacteria are chemosynthetic, which can oxidize compounds like ammonia, nitrate, sulfur, or ferrocene. Green sulfur bacteria, and purple non-sulfur are photosynthetic bacteria. This was chapter 6 first half summary. In the upcoming video we will discuss inshallah the chapter 6 second half summary. Please stay connected, subscribe us and like or comment our channel. Wait for the next video. Allah Hafiz.